Good afternoon, everyone. A warm welcome to this webinar today, organized by HEAL, the Health and Environment Alliance. HEAL is an alliance of over 80 member organizations from the health community in Europe and beyond, working for better health through a healthier environment. And I'm Sandra Jen. I'm the program lead for health and chemicals at HEAL. And today I have the pleasure to welcome Dr. Nikos Stratakis, MSc PhD from the Barcelona Institute for Global Health, ISGlobal, and partner in the EU-funded project called Athlete, aiming to better understand and prevent the effects of environmental hazards on human health from preconception until adolescence. He is a partner in the Athlete project. Dr. Stratakis will provide insights into the links between early life PFAS exposure and childhood metabolic health. Our webinar today is part of a new series of HEAL webinars focusing on different health impacts of PFAS, PFAS being short for per and polyfluoroalkyl substances. PFAS are so-called forever chemicals because they can take up to 1,000 years to break down in the environment, with some not breaking down at all. And the objectives of this new series of webinars organized by HEAL is to raise awareness on the impacts of PFAS, the forever chemicals, and to facilitate access to latest scientific information on the subject. HEAL will use this evidence to advocate for a wide-ranging and health-protective EU restriction of PFAS, which is currently underway. And by preventing PFAS exposure, we know that we can help prevent disease. Finally, a few practical information before handing to Professor Stratakis. The webinar will be recorded, and it is planned to last for about 40 minutes, with sometimes for a few questions at the end. And we will disable the chat during the presentation, and you can ask your questions in the Q&A section. And with this, I hand over the floor to you, Nikos Stratakis.
Thank you very much. And I would like to thank HEAL for the invitation and for the opportunity to discuss such an important issue. So today I'm excited to share some of the latest findings from our research on PFAS exposure and its health impacts. This topic has gained increasing attention due to its widespread implications for public health. Before we dive into the main topic, I would like to start by acknowledging the valuable contributions of our collaborators and funding sources. This is a team science work, and it wouldn't be possible without the dedication and expertise of our research partners and the support that we get from our funders, who also share our commitment to addressing this critical public health issue. So I would like to start with a brief overview of my today's presentation. I will begin with a quick introduction to PFAS, covering some of their properties, exposure routes, and the growing concerns surrounding these forever chemicals. Then I will briefly talk about metabolic health in children, highlighting how early life exposures can have lasting effects on health. And from there, I will present some key research findings from our team on the link between PFAS exposure and metabolic health. Finally, I will conclude with some take-home messages to highlight the main insights and implications of this research. So PFAS, commonly known as forever chemicals, are a family of over 4,000 man-made chemicals that have been widely used in industry and consumer products worldwide since around the 1940s. Due to their unique hydrophobic and oleophobic properties, PFAS are found in several products like non-stick cookware, water-repellent clothing, stain-resistant fabrics, some cosmetics, firefighting foams, and in general various items that resist grease, water, and oils. So PFAS are chemically characterized by their perfluorinated carbon chains, which provide exceptional physical stability and resistance to degradation, making them highly persistent in the environment. As a result, PFAS can be detected in the blood of people and animals globally. Biomonitoring studies from as early as the 2000s reveal nearly universal exposures to several types of PFAS, raising important concerns because PFAS they have a tendency to bioaccumulate in human tissues where they exhibit biological half-lives that can extend up to a decade. So this persistent accumulation underscores their potential for long-term health impacts. The figure up in the right provides an overview of the pathways for human exposure to PFAS. So human exposure to these chemicals occurs through various pathways, including ingestion of contaminated drinking water and seafood, inhalation of indoor air, and contact with other contaminated media. PFAS are widely valued for their non-stick and surface tension lowering properties, making them useful in products that repel oil and water, prevent stains, and in general modify surface chemistry. While direct exposures from consumer products can be reduced through changes in chemical production, long-term exposures persist due to PFAS accumulation in the ocean, marine food chains, and contamination of groundwater. These pathways represent significant ongoing exposure risk, with environmental contamination contributed to human exposure over extended timescales. In a seminal study conducted a couple of years ago, Cousins et al. Compared levels of PFAS measured in rainwater, surface water, and soils with guidelines values. The authors concluded that PFAS defined a new planetary boundary that has been exceeded, based on the PFAS levels in environmental media being ubiquitously above guideline levels. For instance, concentration of PFOA is one of the most widely used and produced PFAS chemical. So concentrations of these chemicals that are measured in rainwater are often much higher than the US Environmental Protection Agency health advisory levels. Notably, even in remote areas such as the Tibetan Plateau, PFAS rainwater concentrations exceed that US EPA limit by approximately 14 times. Drinking water has been identified as a substantial source of PFAS exposure for many populations, particularly those living near to contaminated sites. As you can see in the upper graph, reported by the Environmental Working Group, as of today, more than 2,000 locations in all states in the US are known to have PFAS contamination, including military bases and community water system. And a 2020 analysis of data revealed that PFAS mixture are nearly ubiquitous in surface water, which serves as a primary drinking water source for the majority of the US population. And it is estimated that over 200 million Americans are likely receive water contaminated with PFAS. A recent study conducted by one of the predoctoral students here in our institute examined the spatial distribution of unfiltered tap water PFAS concentrations in the Barcelona metropolitan area. This study not only detected PFAS across the city, but also found relatively higher concentrations in the southern parts of the city, likely due to increase industrial activity in this part. So PFAS exposure in the general population is of major concern. Epidemiological studies have shown that exposure to these chemicals is associated with a range of adverse health effects, including developmental, immune, reproductive, hepatic, and other metabolic complications or the disorders. The focus of today's presentation is on metabolic health. So from here on, I will concentrate specifically on how PFAS exposure impacts metabolic outcomes, especially in children. It is well established now that maintenance of systemic homeostasis and the response to different nutritional and environmental challenges require the coordination of multiple organs and tissues. To respond to these metabolic demands, higher organisms like humans have developed a system of inter-organ communication through which one tissue can affect metabolic pathways in a distant tissue. Dysregulation of these lines of communications contributes to human pathologies, including obesity, diabetes, liver disease, and atherosclerosis. So the term metabolic syndrome was coined to describe a cluster of metabolic risk factors that frequently occur together and increase an individual risk of cardiovascular disease and type 2 diabetes. This condition is characterized by abdominal obesity, high blood pressure, dyslipidemia, and dysglycemia. In parallel with the rising obesity rates that we observe worldwide, metabolic syndrome is becoming increasingly common, and it is estimated to currently affect 1 in 4 adults and 1 in every 10 to 15 children worldwide. However, the pathogenesis of the metabolic syndrome is not fully understood, and significant research efforts are currently focused on identifying modifiable risk factors to help reduce the burden of disease. Already in the 16th century, Leonardo da Vinci's anatomical studies revealed that the developing fetus relies entirely on the maternal environment for proper growth and development. Today, it is well established that environmental conditions experienced during early development can permanently alter an organism's physiology and metabolism, profoundly influencing health and disease in later life. This concept is encapsulated in the developmental origins of health and disease hypothesis, which forms the foundation of much of the work here in our department. In line with this hypothesis, addressing the growing epidemic of obesity and metabolic syndrome requires exploring their developmental origins. This involves identifying preconception, prenatal, and early life exposures that may act as modifiable risk factors for adult diseases. In a recent study, we therefore sought to assess whether exposure to chemical mixtures during pregnancy, including PFAS mixtures, were associated with metabolic risk later in childhood. For this study, we leveraged data from the human early life exposome cohort, one of the most extensive population-based early life European cohorts that involve study sites across Europe. As you can see in this map, participating study sites include France, Greece, Lithuania, Norway, Spain, and the UK. In this cohort, we measured several chemicals in maternal samples and built a composite metabolic syndrome risk scores in childhood using established measure of waist circumference, systolic and diastolic blood pressure, triglycerides, and lipids. Here it is important to note that a higher score is indicative of a higher risk of developing metabolic syndrome. In our analysis, we observed that higher prenatal exposure to PFAS mixture was associated with an increased metabolic syndrome risk in childhood. Notably, maternal PFAS levels in helix were much lower than those typically reported in the US female population. So this suggests that even at relatively low levels of exposure, we might observe measurable effects on metabolic health. Expanding on this, when we incorporated data on proteins and metabolites measured during childhood, we found that prenatal PFAS exposure was linked to higher levels of pro-inflammatory cytokines such as CRP, leptin, interleukin-6, and interleukin-1 beta. Additionally, we observed elevated levels of the branched-chain amino acid, specifically leucine and isoleucine, which are known to play critical roles in glucose and lipid metabolism. Together, these findings suggest that prenatal PFAS exposure has the potential to influence metabolic health well into later life, possibly through inflammatory and other metabolic pathways. Switching now gears, I will focus on the metabolic dysfunction associated steatotic liver disease, which is considered as the hepatic manifestation of the metabolic syndrome. MASLD represent a spectrum of progressive liver disease characterized by excess liver fat accumulation, and recent estimates show that it constitutes a public health crisis, currently affected more than 800 million people worldwide, and almost 1 in every 10 children in Europe and the US. The pathophysiology underlying this condition or the disorder is not completely understood. Recently, it was proposed that the muscle phenotype is a result of bidirectional interactions between the adipose tissue, the liver, and the gut. So multiple hits acting as superimposed insults may be involved in the pathogenesis and progression of the disease, including lipotoxicity of adipose tissue, alterations in gut microbial functions, oxidative stress, insulin resistance, and inflammatory mediators of hepatic and extrahepatic origin. While it is well established that genetic susceptibility, an obesogenic diet, and a sedentary lifestyle are major risk factors in the developing of muscle, emerging evidence now suggests that exposure to certain chemicals may also drive metabolic changes that contribute to fatty liver. These exposures may at least partly contribute to the rapid increase in the incidence of this metabolic disorder. And this concept is known as the metabolism disrupting chemical hypothesis. This hypothesis aligns very well with the developmental origins of health and disease framework and suggests that early life exposure to these chemicals can have lasting effects on metabolic health. This idea is gaining significant attention as now researchers work to better understand and address the environmental contributors to the obesity and metabolic disease epidemics. We recently conducted the first study to systematically review the data on PFAS exposure and liver damage, synthesizing the results of approximately 85 peer-reviewed studies involving rodents. This figure presents an overview of the included studies examining the impact of PFOA on liver weight. The blue symbols represent a significant positive relationship. And as we can see in this figure, overall, it appears that exposure to PFOA in rodents has a consistent positive relation with liver weight across different doses and exposure routes. Moving now to cell culture studies, we recently sought to examine the hepatic metabolic alterations associated with PFAS exposure. We utilized liver spheroids, which are three-dimensional cellular aggregates that are composed primarily of liver cells or hepatocytes, along with other supporting cell types such as endothelial cells, stellate cells, and Kupffer cells. So these spheroids are typically cultured in vitro under conditions that mimic the microenvironment of the liver. And it is important to note that the use of liver spheroids offer a significant advantage in terms of replicating liver physiology, in terms of improving hepatocyte functionality and providing a more clinically relevant model for studying liver biology and related applications. For this experiment, we used multi-donor human liver microtissues to study the impact of PFAS on liver metabolism. This model of liver spheroids is composed of hepatocytes and non-parenchymal cells and is typically used for toxicological studies. So we exposed the liver spheroids with PFHPA for 7 days at a non-cytotoxic dose. And then we evaluated the spheroids for lipid accumulation and using single-cell transcriptomics. We use this specific type of PFAS chemicals, of PFAS chemical, because it has an intermediate carbon chain length and it's structurally similar to PFOA. So PFHPA is highly water soluble and has the potential to aggregate in aqueous solutions while also being highly resistant to degradation. And as a PFOA homologue, PFHPA is assumed to have bioaccumulation potential, binding to proteins in plasma and the liver. And it also has a relatively long elimination half-life in humans of around 3 years. So in this experiment, we observed that exposure to PFHPA resulted in the alteration of more than 400 genes in liver spheroids. Canonical pathways upregulated by this chemical in whole liver spheroids revealed that the majority of these pathways were related to lipid metabolism, while approximately 20% were related to amino acid metabolism. Other important pathways were related to detoxification, energy metabolism, protein trafficking, and hormone metabolism. Canonical pathways upregulated by this chemical in hepatocytes show a similar pattern, with the majority of them upregulated were being related to lipid metabolism and approximately 18% being related to amino acid metabolism. Other important categories or pathways upregulated were energy metabolism and carbohydrate metabolism. Our team conducted the first human study examining hepatotoxicity of prenatal exposure to PFAS mixture, overcoming limitation of the previous few human studies that studied PFAS as single independent compounds. In the real world, in the world that we are living, PFAS occur together as contaminant mixtures in soil, water, and consumer products, are highly correlate, and they may exert synergistic or hepatotoxic effects. The novelty and importance of this work is highlighted by the fact that it was voted as Paper of the Year by the National Institute of Environmental Health Sciences when the paper was published. So for this study, we used data from the multicenter Helix cohort and found that maternal exposure to a mixture of PFAS was associated with a clear dose-response relationship with a higher risk of liver injury in childhood. Children of mothers highly exposed to PFAS, that is to say that those above the 75th percentile, had almost a twofold higher risk for liver injury compared to those of mothers with lower levels of exposure, those below the 25th percentile. And for this analysis, we observed that the results were robust to several sensitivity analyses that we conducted, including further adjustment for maternal fish intake and childhood diet, and we didn't observe any evidence of sex specificity in the observed associations. In the final step of this analysis, we integrated information on serum metabolomic profiling in childhood and used latent unknown clustering with integrated data. This is a novel method which, as its name suggests, is a type of clustering method that aims to identify high-risk groups for liver injury. This analysis identified two subgroups of children. The high-risk group had a 50%, more than 50% increased risk for liver injury compared to the low-risk subgroup. This group was also characterized by increased prenatal PFAS level and decreased serum levels of the branched chain amino acids, valine, leucine, and isoleucine, the aromatic amino acids, tryptophan and phenylalanine, and glycerophospholipids. The results of this study strengthen an emerging body of evidence from previous animals and human studies showing alterations of several amino acid pathways and glycerophospholipid metabolism linked to PFAS exposure. These key metabolic pathways have been also previously linked to liver injury and muscle pathobiology. For instance, the branched chain and aromatic amino acids are associated with hepatic insulin resistance, which is known to be a key pathophysiological feature of muscle, possibly as a result of impaired hepatic tercarboxylic acid cycle metabolism and mitochondrial function. Moreover, abnormal glycerophospholipid levels can induce hepatic lipotoxicity and inflammation. To close, I would like to emphasize a few key take-home messages. First, PFAS exposures are not only widespread, but also chronic, meaning that even modest increases in health risks can result in a significant number of cases at the population level, and this makes it a critical issue for public health. Second, early life exposure to PFAS is particularly concerning due to its potential impact on metabolic and liver health. So, protecting vulnerable groups, especially children, from this exposure is becoming essential. And as we discussed today, addressing this issue requires continuous research and strong international collaboration to be able to unravel the full scope of PFAS health impacts and develop effective solutions. And ultimately, reducing exposure, particularly for sensitive populations like pregnant women and children, should be a public health priority. I hope that these insights have highlighted the importance of addressing PFAS exposure in order to safeguard the health of current and future generations. Thank you again for your time and attention, and I'm happy to take any questions.
Thank you very much, Dr. Stratakis. Really some very important information shared with us and all the participants to this webinar with some, I think, very clear alarm calls for taking actions regarding the chronic and widespread PFAS exposure from these very early moments of life, and sometimes for people who don't even know, women who may not know that they are pregnant. And so it really calls for strong public and regulatory actions on PFAS. I see that we have 3 questions in the Q&A. So one question is about a pregnant woman and the possibility to reduce to reduce the consequences. So I will read the question directly. How long PFAS remain in human body once exposed? Can a pregnant woman reduce metabolites during pregnancy when she applies prevention measures?
Yes, that is a very nice and very relevant question for public health. So, as we said at the beginning, PFAS, they tend to accumulate in human tissues and they have a long half-lives, meaning that they can stay in the human body for very long. I think that one of the most important things for reducing PFAS exposure and is to be aware of them, to be aware of their presence, to be aware of their persistence, and trying to make small steps in our everyday life in order to avoid, let's say, as much as a person can exposure to these chemicals. And I don't remember what was the second part of the question, Sandra.
Can a pregnant woman reduce metabolites during pregnancy when she applies prevention measures?
Yes, the metabolites cannot be reduced per se because and I assume by saying metabolites, they refer to the chemicals. But what we can do is to reduce exposure. And then in the long terms, these chemicals will eliminate it from the body.
Thank you for your answers. We're stressing the importance of trying to reduce exposure by some individual actions, but we know also very much that regulatory decisions to reduce PFAS uses and PFAS exposure are very much the strong answers for the strong response for everybody, because as individuals it's pretty difficult to take these actions, but raising awareness is very important indeed. Another question is, are you also looking into metabolic effects of other PFAS substances such as TFA or PTFE?
Yes, so far the data that are presented from the Helix cohort and the PFAS that we measured in pregnancy, we don't have this data for the pregnant women, but now we are following up the children of these women, and we constantly updating our lists of the measured PFAS chemicals, because you know that although indeed knowledge on the PFAS of the several PFAS, particularly the long chain compounds, they have brought an incentive for phasing out of these chemicals. However, following this, several other compounds, including the shorter chain PFAS or the so-called PFAS alternative, are being constantly introduced, have been adopted as a new methods by the industry and for other uses, and are now increasingly being introduced in the environment. So our list is constantly being updated in order to be able to measure not only the legacy PFAS compound, but also the new ones.
Thank you for this comprehensive answer as well to that question, and this constant updating of the topic. Then there's another question. Well, I think you partly responded to it with the links between the legacy PFAS and the newer one. Most of presented results are related to legacy PFAS, such as PFOA. Is there any available literature evidence for the effects of short chain and emerging PFAS on obesity and metabolic syndrome?
Yes, and so far most of the evidence comes from animal studies. And as I'm saying that now our team and many teams around the world are updating the list of measured chemicals in order to be able to measure and examine the health effects of the newly emerged chemicals. But it is important to know that this sometimes takes a lot of time. So what we wrote, Professor Martin Vrijheid, who is an expert on this topic, and we recently wrote in an editorial that unfortunately the pace of our research is not fast enough to address the rapidly changing chemical landscape and the emerging concerns of these affected populations. So that's why we need to be fast and we need very strong collaborations in order to be able to let's say match up the rates of how new chemicals are entering the environment.
Thank you. And maybe also a plea for applying the precautionary and prevention principles in the regulatory approaches. So I will, if you allow me, I will propose that we take a last question.
Yes.
um, so it's about your reference to the planetary boundary. And the question is, are you talking about the one made by the Stockholm Research Center? And in that case, was it made measuring only PFAS?
I think that in this study, it is a seminal study indicating a very important problem that we can measure PFAS everywhere. Uh, I think that in this study they assessed four different types of PFAS, four different types of legacy compounds. It was done by this team of researchers, but I was not involved in this research, so I'm not. So I suggest that to go in the suggested reference and read more about it.
Thank you. Well, thank you very much, Professor Stratakis, for this extensive presentation on the links, on the insights between PFAS exposure, early life exposure to PFAS and childhood metabolic health. We will very much, of course, as Hill, use all this information and for our work on making sure we have a comprehensive and strong restriction at EU level on PFAS and for all the other complementary actions that can be taken. And with this, I also like to mention the next HEAL webinar on PFAS that will be focusing on PFAS and the links to certain types of cancer. And I'd like to thank you very much again for your time and your availability to answer all these questions. And with that, I will say goodbye to everyone who joined us today. Thank you.
Thank you all
Goodbye.
very much. Thank you.
Thanks.